Hi everyone, my name is Staron. This is the next episode of MedChem Essentials, scientific seminars devoted to medicinal chemistry. Today we continue talking about plasma protein binding. Today's presentation will start with the free drug concentration, then we'll see the relationship between ppb and hepatic and total clearance, then we'll move on to oral bioavailability, and finally we'll conclude overall information regarding plasma protein binding. Now, I would like to present something that when I first looked at it, it was kind of surprising to me. But after second thought, it became quite clear and logic to me that despite the level of protein binding, we will finally see the same free drug concentration. Why is that? Because if we have high protein binding, then larger fraction of the drug will be bound to the proteins and lower fraction of the drug will be free-flowing in the plasma. Now, when it comes to the clearance, it's only the free-flowing unbound drug is unable to clearance, whatever clearance, is unable to be cleared from the body. So if we have low free drug concentration, we will have lower clearance of the drug. Now, in the situation when we have low protein binding, we have higher fraction, higher concentration of the free drug in the plasma, and because of this higher concentration, we'll observe higher clearance of the drug. So this drug will be cleared much quicker than the drug that has high protein binding. And finally, we will see same free drug concentration, but with one caveat, with one observation that in the upper case, we will see higher total drug concentration than in the case of the situation when we have lower protein binding. So in the case of high protein binding, the bound drug functions as a sort of a reservoir, refreshing and bringing up the drug that was cleared from the body. How this impacts hepatic total clearance? So in the total clearance of a drug is dependent on the hepatic blood flow, is dependent on the ppb as described on the previous slide, and is also dependent on the intrinsic clearance. We will have a separate talk on the clearance in general, so no worries if you don't understand these terms. The intrinsic clearance is the intrinsic property of a substance, how quickly it is being metabolized by, in this case, hepatocytes. So finally, it's the information is that the lower the ppb, the higher the total clearance becomes, and this very nicely relates to what we showed on the previous slide. So if we observe an increased clearance of a compound, it might be due to reduced ppb and not increased metabolic rate. So when we compare two compounds and they have different metabolic rate, it might not necessarily be due to the one drug being more prone to metabolism and possessing more metabolic hotspots, but it may be due to reduced ppb. It is very important to remember about it when we compare in vivo clearance data for one drug, one compound to another, when their ppb is much different. Now let's see at the oral bioavailability. It is determined by, again, hepatic blood flow. The reminding that oral bioavailability F is the fraction of the drug that is absorbed into the system, fraction of a dose administered. So it is determined by hepatic blood flow because the administered drugs they travel through the liver and can be metabolized during this first passage. It's dependent on the ppb and is dependent on the hepatic intrinsic clearance. So in the end, the lower the ppb, the lower the oral bioavailability. In general, most often you can find that low bioavailability is associated with low solubility, low permeability, or high metabolic stability. But sometimes it's the low ppb that can lead to lower oral bioavailability. Now let's have another example. There is I will give a statement that there is no gain in reducing ppb without reducing the intrinsic clearance. And why is that? In the in vivo study of the following compound, which was tested in 2 strains of rats, CD and SD rats, this compound exhibited only a fraction of, the fraction unbound in CD rats was just the 1/10 of that in SD rats. As a consequence, the total drug concentration was 6-fold lower in case of intravenous administration and 19-fold lower than in the oral administration. But despite that, the unbound concentration of this drug remained the same in both strains of rats. Now, I was telling you previously how ppb is important, how it determines and affects multiple parameters of drug properties. Now I will tell you why ppb is not that much relevant in terms of drug design and development. It is a fact that clinical drugs, they have rather high ppb. Majority of them actually exhibit above 90% of plasma protein binding. One of the example is sertraline. The unbound fraction of sertraline is really low, and it didn't prohibit this compound from being the best-selling antidepressant in the United States. And in the same time, the unbound fraction for meprobamate, which was one of the best-selling drugs in the 50s, was 0.76. So the low unbound fraction for sertraline and rather high unbound fraction for meprobamate, it didn't matter, and both drugs made it to the market and were and are one of the best-selling drugs. So, concluding, ppb should be measured in the following cases. Firstly, it should be determined to investigate the potency shift if the potency shift between assays is due to ppb. Second of all, it should be determined in the pharmacokinetic and pharmacodynamic studies to assess the relationship between in vivo free concentrations and in vitro free IC50s. Also, when we have a CNS drug, it should be determined, the ppb should be determined for the brain tissue and for the peripheral tissue to calculate the unbound brain to unbound plasma concentration ratio in the brain. Also, in the human clearance prediction studies, it should be determined to calculate based on the intrinsic clearance from microsomes or hepatocytes to calculate the predicted human clearance. But in the end, ppb should not be a matter of structure optimization. For the first instance, it's kind of challenging to optimize ppb independent of all other parameters. Second of all, even very high ppb does not prohibit many of the drugs to be introduced into the market and be one of the best selling drugs. Thank you very much for your attention. See you next time. Bye bye.